Hi everyone, Dr. Ron Bauer here to explain some of the principles involved in Bio-Rad's pGLO Bacterial Transformation Lab, as well as to review the basic protocol and what to expect when you're doing this lab. So essentially what we're doing is taking a genetically engineered plasmid and transforming bacteria with it through a heat shock protocol. We can then use the principles of the scientific method, use our scientific reasoning to show how a bacterium can take up a plasmid and express the genes in that plasmid and exhibit the traits created by the proteins generated. So it's really an important, fun lab to reinforce or perhaps teach gene expression to your students. So basically what we're seeing here is the act of transformation, taking a plasmid, putting it into a subset of bacteria, growing up that bacteria, and then making those bacteria express the gene, make a protein, eventually show this trait, and in this case it's glowing. So it's a fun lab. The pGLO plasmid shown here, so plasmids, as a review, are these extra chromosomal loops of DNA, and they have genes on them, so you have your large chromosomal loop and your small plasmids. So basically you see to the right here a map of Bio-Rad's pGLO plasmid, and there's some key features here. So as you might recall from lessons on DNA replication, basically DNA replication starts at an origin of replication, and so that's what we have here. This ensures that the bacteria plasmid is copied into each clone during binary fission as the bacteria multiply, so you get copy after copy after copy of the plasmid in DNA for further expression. The BLA gene, shown here on the plasmid, shown in red, encodes the enzyme beta-lactamase, which is an effective, very effective antibiotic. This enzyme basically works by hydrolyzing or breaking down a certain class of antibiotics, including ampicillin, which is what we use in this lab. So it basically breaks that antibiotic down. So essentially what you're doing is by giving bacteria this gene, you're conferring the trait of antibiotic resistance to any bacterium that possesses that plasmid. The next facet of this map I want to direct you to is the RSC gene. Okay, here shown in blue. Basically, this is the Arabinose operon regulator. So you're going to see here in a moment what Bio-Rad has done to utilize the Arabinose operon. But basically what you have here is the gene that encodes the regulator that can control gene expression. Okay, so basically the regulator blocks downstream expression of a gene in a certain conformation, and that conformation can be changed when you add an inducer, which we'll get to here in a moment. Last but not least, the star of the show, I want to guide your attention to GFP. So it encodes the jellyfish green fluorescent protein. So GFP in this case is downstream of the Arabinose promoter, so it's under the control of that promoter, which can be blocked by the RSC protein, the regulator. So we're going to be able to induce expression of this downstream GFP gene with the sugar Arabinose. Next, what we have here is a little depiction of certain elements of the Arabinose operon and what Bio-Rad has done to utilize this operon. Okay, so the molecular biologists at Bio-Rad have put this Arabinose operon to use to regulate gene expression. The reason I love this lab so much is it really reinforces that regulation, that prokaryotic regulation that the operon affords. And operon itself is a concept that has a lot of different facets to it, so I really like it. In fact, molecular biology is founded on the fact that DNA and genes are modular in nature. That is to say that they can be transferred between organisms and they behave the same way in each organism. So the genes can be expressed equally the same in both organisms. And for AP biology students out there, that's a really key part of Big Idea 1, really integral idea for evolution. So basically what we're seeing in this picture, in this first line, GFP, the GFP gene downstream of the Arabinose operon. So we're regulating it. So RSC, that regulator protein in this particular conformation, blocks the promoter. So you can't even see the promoter in this because it's blocked by that regulatory protein. However, if you add Arabinose, which I'm going to depict as this, if you add Arabinose, you structurally change, so the conformation of ARA changes such that now you can actually see the operon promoter there, so the promoter for those BAD genes. And basically now what can happen here in line 3 is that RNA polymerase can come to the Arabinose promoter and start expression, right? So basically you open up the promoter so that RNA polymerase can bind there and start transcription. And that's basically what we're seeing here in line 4, is that the RNA polymerase has proceeded and it has transcribed this GFP gene here into messenger RNA, which will eventually be translated next into that GFP protein. So a really interesting lab here. Okay, so what we'll talk about next here is some of the AP biology surrounding ways that bacteria diversify their genomes. There are other screencasts that talk about transduction and conjugation as ways for bacteria to diversify their genomes. I'm going to focus here on transformation. Okay, so transformation is an important one. We're using it, in fact, for the lab. And what transformation is organisms have the ability to be transformed. Let's see here, get my eraser. So when this bacterium here, if I can erase this little segment right here. For example, if this bacterium were to open, say the bacterium died and the cell wall started to collapse, these plasmids could come out, and what would happen is the genes that are on these plasmids, if they're able to be transformed, that is to come into this second bacterium here, the genes on that plasmid will be expressed the same as any plasmid that's already there, and also the same as the large chromosomal loop. So we're going to take advantage of this ability for bacteria to be transformed in our Biorad pGLO transformation lab today. Okay, so let's introduce you to the actual start of the pGLO transformation experiment here. You could use this as a protocol. Of course, your teacher will give you a protocol to follow, but here's basically what's going to happen. You're going to designate 2 different tubes, one as minus pGLO, a tube that will not receive the pGLO plasmid, and one as positive pGLO that will receive the plasmid. Now both tubes, are going to get colonies of bacteria from that starter plate, okay? So you will have grown up or be given a starter plate of bacteria. The plate contains individual colonies, which you'll use for your research. You're going to add these colonies to the transformation solution used in the kit, the calcium chloride. And that's how we start. So you're going to have bacteria in both tubes. Now, one of those tubes, the plus pGLO tube, is also going to receive pGLO plasmid. So you'll also have a tube of that to add to the plus pGLO experiment. Okay. Next, we're going to take those bacteria, half of which have been exposed to plasmid, the other tube is not exposed to plasmid, and we're going to put them both through a heat shock protocol. So a heat shock protocol is a form of transformation. So to really shock the bacteria, you go from ice to heat for a brief time, and then you put them back on ice. And it's really important to stick to this timeline of 50 seconds in the 42 degrees Celsius water bath, and that is because the bacteria are being shocked. Many of the bacteria will die in this process, certainly more than you want if you extend the protocol past 50 seconds. Okay? The thing is, though, is that many of those bacteria will also live, and an even smaller subset of those bacteria will have been transformed. That is to say, they will have taken up the plasmid provided to them. Okay? So basically, you're going to do 10 minutes on ice. So you'll have your tubes here. You'll transfer them for 50 seconds to a water bath of 42 degrees Celsius, and then back onto ice. Okay? Next, the two tubes will receive what is called LB, essentially food nutrients for the bacteria. So after your incubation, you will add LB. So each tube will get some LB. And essentially what you're doing there is you're saving the survivors. Okay, you want them to start gene expression and basically start enabling themselves to divide and express genes to make proteins. Okay. So let's get into basically how you're setting up your experiment. Okay, we're going to put our various minus pGLO samples and plus pGLO samples onto different plates. Okay, so you're going to have an LB plate. Basically, that's a gel matrix with food to grow on. You're going to have two LB ampicillin plates. These plates are a gel matrix with food, but it also has the antibiotic ampicillin. So keep in mind that antibiotics kill bacteria. So you'll get two of those plates. And last but not least, you'll get a plate that has all the above, but also Arabinose. So you'll get an LB ampicillin plate, food, antibiotics, but you'll also have that inducible Arabinose sugar. Okay, so you're going to have a plate that can induce expression of that downstream GFP gene. Okay, so let's walk through this a little further and see what we can get out of the experiment. So essentially what you'll do is you'll add a sample of minus pGLO to your LB plate. And this can be seen as essentially a positive control. Both of these are going to serve as controls. And keep in mind, a control is something you compare your experiment to for various reasons. But essentially, you're gonna compare your controls to your experimental samples to validate your experimental results. Okay. So essentially, adding heat-shocked bacteria to an LB plate, which contains food, it's going to serve as a positive control for us, okay? So they have not received plasmid, they have received a heat shock, and we do this control for several reasons. It lets us know if bacteria can survive a heat shock, okay? So that might be a question that can be solved by this negative control. So if this plate doesn't have bacteria, then one would predict that none of the other three plates would have bacteria. So this is basically telling us if we have bacteria that can survive heat shock and that they were alive and viable to start. Okay. The other control, the LB ampicillin plate that receives bacteria that were heat shocked but not in the presence of plasmid, will serve as this negative control. Okay, this is one that you would predict the death of all the bacteria, given the fact that this plate contains antibiotic, but the bacteria that were plated on it that were heat shocked in the absence of pGLO. So these bacteria never really had a chance to be transformed and obtain that ampicillin resistance gene, the BLA gene. Okay. Now, when you get your results for this particular plate, you're going to ask yourself, well, they should all be dead. If they are alive, did you have bacteria that were naturally resistant to ampicillin? That would be a problem for this experiment. Or you could call into question if your ampicillin is actually working. So do you have functioning antibiotic that can kill bacteria. If they're growing, maybe not. Okay, now let's transition into our experimental samples. Okay, essentially what you're going to do is you're going to take samples from that plus pGLO and plate them onto the other LB ampicillin plate. So again, there's food here, but there's also antibiotics on this plate. They will receive heat shock in the presence of pGLO plasmid. So they have the opportunity for transformation. And essentially what we're doing on this plate is we're checking for those transformant bacteria using antibiotics. So it's a means of selection. The biological reasoning here is that the only way that you can live on a plate containing ampicillin is if you have that BLA gene. Okay, were you transformed? Do you have the BLA gene? We're asking these questions for bacteria. And if they're living on this plate, most likely they do have that gene, and they have obtained this trait of resistance. So we're checking for transformation via resistance to ampicillin. Okay. The last plate here is the fun plate. It is the LB ampicillin Arabinose plate. And essentially, yeah, we're confirming again resistance to ampicillin, but we're also checking for expression of GFP. That's the whole purpose of this experiment, to see if we can take that jellyfish protein and express it in bacteria. Okay, so remember, Arabinose is the inducer for the Arabinose operon, and it basically opens up the promoter for expression of downstream genes. So on this plate, we're checking to see if we have induced expression of GFP with Arabinose. So before we advance to the next slide, I want you to pause the video and hypothesize what you would see on each plate given the experimental setup. This is sort of explaining the controls, the experiments, where everything goes, but I want you to make sure that you understand the lab and are able to predict what you'll see experimentally. When you've done that, go ahead and press play, and you can rejoin us on next for the data discussion part of this particular video. So these are the results that one would receive in this experiment if everything goes well. So let's take a look at this LB plate first. This plate received heat-shocked bacteria. You would see a lot of bacteria just because this is a growth plate. There's no antibiotics. Sure, some of the bacteria died in the heat shock process, but keep in mind that colonies contain millions of bacteria. There are so many bacteria, in fact, that you would see what we call a lawn here. And a lawn is essentially, you can see from the shading, but you're not going to see colonies. You're just going to see loads of bacteria. It's going to be basically covering the entire plate. Next, on the LB ampicillin plate, you shouldn't see anything on this plate. There should be no growth whatsoever. You plated bacteria that were not exposed to plasmid during heat shock. So yes, they were heat shocked. Some of them died for that reason, but all of them died. All the survivors died because they were exposed to antibiotic. They did not have the plasmid, so they could not be transformed. They could not take up the BLA gene and express an antibiotic resistance protein. So they were not able to obtain the trait of resistance. Everything is dead there. It should be a clean plate. The second LB amp plate here, it received bacteria that was exposed to plasmid during heat shock, so it should have some bacteria that were transformed and received the BLA gene. Thus, you would see the trait of antibiotic resistance in the form of colonies. Each colony represents a bacterium that was transformed. You do not see a lawn, however, in this case, because many bacteria were not transformed and consequently died because of the exposure to the antibiotic ampicillin. But each of these colonies here was an individual bacterium that was transformed and thus can survive in the presence of antibiotics. So you have some colonies here. Last but not least, let's take a look at our LB amp Arabinose plate. This plate received bacteria that was heat shocked in the presence of plasmid like the previous one. So you have antibiotic resistant transformants. You would also have Arabinose sugar on this plate. Remember, Arabinose is the inducer, so it takes the regulator protein and changes its conformation such that RNA polymerase can come to the promoter and start expression. So that downstream GFP gene is transcribed and translated, resulting in colonies expressing GFP, colonies that grow. This plate over here, although they lived, no glowing. They are not inducing expression of GFP. So if you shine a UV light on this one, there's no glowing. If you shine a UV light on this one, you do get glowing. Okay, so that's really the basics of this BioRed pGLO GFP expression kit. I think it's fascinating. I think it's a really fun lab. It reinforces a lot of concepts in AP biology. It's a great lab. And this is what you would see at the very end using a UV pen light on the LB AMP Arabinose plate. These are essentially bacteria that are expressing a jellyfish protein called GFP. That brings into focus a lot of important ideas, certainly a lot of from Big Idea 1 and Big Idea 3 in AP biology. Anyway, I hope this ties it in nicely. I hope it gives you a really nice mental picture of the lab and some of the expectations for it and some of the results that you'll get. Okay? Let me know how it went. See you next time.